Maybe when you're ready to start. Screen sharing. So that should be fine now. So. We'll get started, guys. It's my great pleasure to introduce you to Dr. Louisa Fitzpatrick today from, from the Behavioural and Evolutionary Ecology Research Group at UTAS. It happens to be the same group I did my PhD in, and I was very lucky to have Lou as a friend and mentor during my PhD. We shared an office, we shared a supervisor, we shared a study species. I mean, yeah, what more could you want? So Lou originally hails from WA. She did her undergrad and honours at University of Western Australia, working on sperm competition in guppies for her honours. So really interesting topic. She's also done a bit of work as an environmental scientist and an education assistant for the WA Museum, and then came to Tasmania in 2015 to do her PhD with Professor Eric Wopstra and Associate Professor Jeff Weil. And she also was the recipient of an Endeavour Scholarship, got to travel to Sweden and work over there and do some interesting experiments in Sweden. Not to mention it was Sweden. I mean, pretty awesome. And as I said, she's been a great friend and mentor of mine, and I'm very excited to hear her talk today on senescence in a skink. So we'll hand it over to Louisa.
Thank you, Peter. All right. Hello, everyone. I'd like to acknowledge before I start the Palawa people of Lutruwita, who are the traditional custodians of the land where I do my research. And as Peter said, I'm talking about senescence in lizards, which is aging, and I'll be talking broadly about the work that I did during my PhD, and some of that is continuing now, and then what my sort of future ideas are. So I'm still working in the same area. So senescence is aging, it's biological aging, which is associated with a decline in function and an increase in mortality. So the kind of things that we know visually, grey hair, wrinkles, those type of things, it occurs in all animals and plants, even in natural systems where it wasn't picked up on for a while because things are preyed upon. So they often don't reach extreme old age. And we know patterns of senescence vary a lot between species, between populations, between individuals. So one person might look amazing at age 70, someone else might look terrible at age 20. Some animals live for a year, some live for up to 200 years. So it's really interesting, skipping ahead, really interesting to think about the variation in aging. And it's one of the fundamental questions in biology. And so aging can take place at an organism level, like those physical changes you see. It's also underpinned at a cellular level. When I talk about aging at a cellular level, I'm talking about telomeres usually. So telomeres are a bit more popular now, you might have heard of them. They're little caps on the end of DNA that protect the important coding information from being eroded as the cells divide. So every time cells divide, a little bit of the end of the chromosomes are lost, and the telomeres are just a bit at the end, like a ferrule on the end of a shoelace or the sole of a shoe that gets worn away instead of the important information. So when telomeres get too short, it leads to tissue death, organ failure, and then it can actually lead to organism death in that sort of pathway. So the importance of telomeres and the enzymes that repair them were discovered in 2009. There was a Nobel Prize won by a Tasmanian researcher and her associates. And traditionally, they've been really studied in biomedical research because obviously we care about us and how we work. And they're linked to longevity and age, biological age of individuals, but the causal links have been heavily debated and they're still not really understood. Does shorter telomeres cause aging, or is it a sign of aging? And there's been a lot more work lately looking at telomeres as markers of age. So markers of the biological age or condition of an individual within a population. And telomere length is not a static quality. So it's in constant flux. Telomeres are shortened by cell division, as I said before, and also by oxidative stress, as the metabolism, oxidative stress is a product of aerobic metabolism. So that attacks the guanine-rich sequences of telomeres. But then telomeres can also be repaired by telomerase, which is an enzyme that rebuilds the sequences, and antioxidant activity can protect against the oxidative stress. So more of one thing leads to telomere loss, more of the other leads to telomere repair. And the enzyme telomerase, which repairs telomeres, is actually suppressed in endotherms, so birds and mammals and us. Once we are born, you have it in the embryonic stage and then it's suppressed. So we actually don't have that available to repair our telomeres, but it is present in ectotherms. And because these things are in a balance, it leads to the kind of trade-off, classic life history trade-off, whether an organism invests in growth and reproduction, which comes at a cost to telomere length, or do they invest in somatic maintenance in terms of repairing the telomeres, looking after them, making sure that they can survive for a longer time. So I wanted to understand what influences telomere length. And we know that it's heritable. So telomere length from juveniles to, from adults to juveniles can be inherited. Also from juveniles to adults, it changes. So either it gets shorter or it could be repaired. And this is influenced by the life history that an individual experiences. Telomeres can also be influenced by the environment. And from endothermic research and cell line research, we know that a whole host of things can influence telomere length quite drastically. So all of the things up on the board, I won't read them all out, but sex, size, parental care, climate, pollution, all of these things can have varying effects on telomere length. And these things all differ between species populations and life history stages. But ectotherms have been less studied in this respect, and they're 99% of species worldwide. Most of the research is done on endotherms. But ectotherms are really interesting in terms of aging and telomeres because they're so different from endotherms. So they have indeterminate growth, or some have asymptotic growth. So they keep growing past their reproductive maturity, unlike us and mammals and birds. They have dramatic metabolic shifts. So they experience a lot larger variation in temperature compared to endotherms, which maintain a stable temperature. They also have somatic telomerase present. As I said before, they keep that enzyme going that can repair telomeres if needed. So the patterns of aging aren't well understood. So there's things like turtles, which you're probably aware of, can live for hundreds of years. But how do they do that? And why is it different from endotherms? And to study these questions, I used the little spotted snow skink from Tasmania. It's a small, about 10 centimeters long lizard, and they live for up to 14 years, generally not that long, but that's the longest one we've observed. And this system is really cool because we have 23 years of longitudinal data on the females. So every year we catch all the females from two populations, we bring them in, measure them, they have their babies, we measure all the babies and we put them all back. So we know individuals year to year and their reproductive history. And there, as I said, there's two populations. We have a warm lowland population, which is down at sea level and it's in kind of bushy areas. And then we also have a cool highland population, which is up in the central plateau of Tasmania, where it's covered in snow for all of winter. So they're quite different climates. And the difference in climates has led to a divergence in life history. So the ones in the cool highland get bigger and produce more babies, basically. So these are the four broad topics of my thesis. There were two experimental data chapters, the temperature in telomeres and tissue regeneration in telomeres, where I did experiments. And then the second two chapters, I used the long-term data that we have to look at questions about organism and population level senescence rather than the cellular, which was the experimental work. So to start with, how does temperature affect telomeres in an ectotherm? So we know that higher temperatures speed up metabolic rate. And in ectotherms, we'd expect this to be a really strong relationship because their metabolic rate or their body temperature is directly related to the environmental temperature. A faster metabolic rate creates more oxidative stress and more cell division, which in turn should increase the telomere attrition. Telomeres should be getting shorter in hotter environments. And I also expected there to be some level of local adaptation. As I said, I have those two populations. So in the cold environment and the warm environment, they should be adapted to the environment they're living in. So what I did was to take two populations in the cold environment and two populations in the warm environment. And I put lizards in hot thermal treatments and cold thermal treatments for 3 months. I took blood samples at the start and the end of that 3-month period, and I looked at the telomere length and the oxidative stress levels in the lizards. And what I expected was that we would see faster telomere attrition in the hot treatment. As I explained, the metabolic rate should increase, and particularly for the cold climate lizards, because they would be adapted to the colder climate. What I actually saw was that telomeres got longer in the hot thermal treatment. So this is not what I expected. And there was no local adaptation. The two sites were the same in terms of the telomere length. And this suggests to me that telomerase activity is present. The telomeres are getting longer from the start to the end, so something must be repairing them. Which puts a different view on the current kind of idea about climate change in telomeres, which has been sort of on the vein of, with climate change, hotter temperatures will mean faster telomere attrition in ectotherms particularly. So this needs a little bit more investigation. And the oxidative stress changes were even more complex and unexpected. They were site-specific, so it's a bit confusing on the graph, but the highland is the cold population. So we saw an increase in oxidative stress when the cold population were put in cold temperature treatments compared to the cold population in hot temperature treatments. So that's also not what we'd expect. That suggests to me that there's some antioxidant activity going on in the warm population, kind of keeping those stable. And it's interesting that the hot treatment in the cold environment didn't increase stress. Maybe they appreciated the warmer temperatures and they were able to function better. What does this mean in terms of the balance of telomeres? Like I said, measuring telomere length, it's dynamic. So each of the cellular processes that are operating are actually up and down themselves. So each of these things could be happening at a different thermal optima and at a different time scale. So it's a lot trickier than initially I thought when I started my PhD. And the telomere length that I measured is just a snapshot at one time. So any of those processes could be at any point in time, in temperature. And that really just led me to think that telomere processes at the moment are a black box for me. I know what went in, I know what came out, but I need to discover more of what was going on internally and measure those processes concurrently at different timescales. So then I did another tissue, another telomere experiment at the same time as that one. Otherwise, I would have added some more measures in, looking at tissue regeneration. So ectotherms are really interesting because they can regrow entire limbs. So there's been a little bit of work done on fish and marine invertebrates about how that affects their telomere dynamics. You'd expect that such large scale tissue regeneration would lead to huge amounts of cell division and the telomeres should get shorter. And this is partly seen. There's been some studies where at old age, fish are less able to regenerate tissue than at younger ages. So it might be context dependent as well. So what I did in this experiment was I took blood samples from lizards and then I removed tails from half of them and let them regrow those tails, and then took a blood sample again after 2 months of either regrowth or just control. And I measured again the telomere length and the oxidative stress in these individuals. And I expected telomere length to decrease as the tissue was regenerating and to stay the same in the control group. And I expected oxidative stress to increase as well because it's a stressful thing for the individual to regrow such a large portion of their body. It takes a lot of resources. What I actually found was that telomere length increased in the control group and stayed around the same in the group that were regenerating their tails. So this suggests to me that telomerase activity is present again, and the control group are maybe making use of the beneficial experimental conditions. They were outside in little tubs, they were protected from predators, and they were getting fed regularly. So maybe they're just enjoying the scenario there. And the oxidative stress decreased in the regenerating group, which is completely what I didn't expect, and stayed around the same in the control group. And this was an interesting result because you expect them to be more stressed, but it's possible that they're counteracting that stress by upregulating their antioxidant activity. And possibly the timescales that I was looking at were the wrong timescales to see the changes. And the implications from this study, we know that cellular maintenance versus cellular division is at play in these animals. They're investing in maintenance in the control group because they don't have to invest in regrowth. And tail loss can have significant long-term consequences because the lizards that were regrowing their tails didn't get the opportunity to invest in their telomere length for the long-term goals. And so the consequences of tail loss may affect longevity in lizards. It might shorten their lifespan, but it might not because we don't actually know if telomere length is related to lifespan in the lizards yet. So then for the third chapter, I wanted to have a look at that, see if life history was related to telomeres in the species. So from the previous 2 chapters, found that tail loss influenced life history, which has an effect on the telomere length in the adults, and temperature has a direct effect on telomere length. Then I wanted to look at the cumulative effect over a lifetime for these lizards. I wanted to look at the life history patterns. So what are juvenile experiences from birth to adulthood, how that influences their telomere length, and also the genetic inheritance. So does a mother's telomere length translate to an offspring telomere length? We only have females in this population, so we can only look at the mothers to offspring. And then also to look at the environmental effects as we have the 2 populations with different life histories. So I looked at 3 factors in the juvenile to adult stage, looked at age of individuals, the growth, so their body size, and reproductive investment. So the most recent time they reproduced, how much effort they put into it. And then from the juvenile side, I looked at embryo development, so their birth date, which is a proxy for their developmental time or speed in the species, and the mother's age to see if there was a maternal age at conception effect. As in humans, it's quite In humans, it's understood that older mothers give birth to offspring with shorter telomeres, but older fathers give birth to offspring with longer telomeres. So it's, again, very complicated and individual. So for this experiment, there's two parts: the changes over life, which is from birth to adulthood, and then the changes, the inheritance. So how a mother's telomere length and other factors influence the offspring telomere length at birth. And I did each of these in 2 sites, and I think there were around 20, 25 samples in each group from that long-term population study. I measured the telomeres and found that the results did differ between the populations. So environment had a significant effect on the life history and how it related to telomeres in the species. So in the cool highland population, juvenile telomere length was the strongest predictor of adult telomere length, which is not unexpected. We also found that pattern in the warm lowland population. In the warm lowland population, there was also an effect, there was an interaction effect of age and growth on the adult telomere length, but the effect size was very small. So take that with a pinch of salt. And there was no effect of reproductive investment on adult telomere length. So whether they invested heavily or not very heavily didn't have an effect in the short term. So in both populations, the main predictor of adult telomere length was juvenile telomere length. And there was a difference in the average telomere length between the 2 sites. So if juvenile telomere length is the most important thing predicting telomere length at adulthood, what influences telomere length at birth? In the warm population, we found that the mother's telomere length had a significant effect on the juvenile telomere length. So it could be maternal effects or heritability. I need to do some further studies to unpick those things. And there was also some significant but small effect sizes, again, effects of the birth date, so the speed at which the offspring are developing, and the mother's age. So older mothers actually gave birth to offspring with slightly longer telomeres, so counter to what happens in endotherms as well, generally. And these effects were not present in the cool highland population, so there was some specificity. So this tells us that there are clear differences between the 2 sites. There's an impact of environment, or at least of population, on the way these things play out, the telomere dynamics. Genetics is important, probably, because telomere length at birth predicts it throughout age, throughout life. And there is some interaction going on between maternal age and the site specificity. It's interesting to think about it in terms of what we know from endotherms. So in the lizards, the telomere length actually increases with age, which is not what we see in most birds and mammals. So this makes sense, considering we expect there to be telomeres present and active. There's no strong size effect on telomere length, which is counter to what we expected, because as you grow more, there's more cell divisions. But again, maybe they're accounting for that by upregulating telomeres. And the fact that we didn't see any proximate effects of reproduction was interesting, because we know that reproduction is costly, particularly to females, and investing different amounts should be visible at some level. So then I wanted to have a look at a broader view, taking a look at the population level and at general patterns of aging and senescence in the 2 sites. So the pace and shape of senescence is influenced by intrinsic factors and extrinsic factors that mediate the trade-offs, life history trade-offs between investment in growth, reproduction, and maintenance. So intrinsic factors, individuals optimize their fitness for the average lifespan the species experiences, and to individual cues about how much lifetime is remaining to them, to try and maximize their fitness. So the amount of offspring they're passing on to the next generation, amount of genes. And then there's also constraints from extrinsic factors, which could be direct constraints like predation or resource availability, or indirect constraints through the way that the environment influences life history in the animals. So those two things are playing into the trade-offs. The individuals are making decisions about allocation to growth, reproduction, and maintenance. And these decisions play into the pace and shape of senescence. When I talk about pace and shape, that might not make sense because you think aging happens at the time that animals age, but senescence is really the biological age. So thinking about demographic senescence, it's measured by mortality changes with age and fertility changes with age. So there's different patterns. There's positive senescence, which is what humans have. Our mortality increases with age and our fertility decreases with age after maturity. But in some animals, we see negative senescence, often in ectotherms, where mortality actually decreases with age and fertility increases. If you think about a snake, it gets bigger and bigger and bigger as it keeps growing, and then it can have more and more babies. And if there's no mortality increases, then it's not being selected for to age necessarily, it's being selected for just to keep growing and keep having babies. There's also a pattern of negligible senescence where mortality and fertility don't really change with age. So they're the three kind of things that I was looking at to see which one of these was operating in the lizard species that I look at. And of course, these things don't happen in a straight line all the time. They can be curved. So for example, this is humans, the graph, yearly probability of birth in males and females with age. So you can see it increases up to the age of maturity, and then after maturity decreases. And that's also the pattern we see in mortality. It starts to increase after maturity. But ectotherms have a much broader range of senescence patterns available to them than this kind of increase and decrease. As I've said many times already now, they can repair their telomeres throughout life. So they have that enzyme present that enables them to repair their telomeres. And their continued growth after reproductive maturity can select for that negligible or negative senescence because their reproductive output is actually increasing with age. The variation in senescence in ectotherms can be really useful because it doesn't necessarily fit the theories, the evolutionary theories of aging that we have currently. It raises some questions about these. So again, to remind you, I was using the snow skink, which is really great for this study particularly because to study demographic senescence, you need long-term mark recapture studies. And those are really rare in ectotherms, particularly in terrestrial ectotherms. So I'm very lucky to have this data set available to me to be able to ask these high-powered questions. And again, the lizards have asymptotic growth and life history differences between the two populations. So I can look at demographic senescence in the two populations to see if life history informs the patterns of aging. So when I talk about asymptotic growth, this is what I mean. This is a growth curve for the two populations, the blue cool highland and the red warm lowland. And you can see that the cold highland lizards do grow larger overall. They also produce more offspring, we know. There's no apparent difference in the maximum lifespan between the two populations. They both live for around 13 to 14 years as a maximum. And I wanted to know if there is either actuarial senescence, an increase in mortality with age, or reproductive senescence, a decrease in fertility with age in the species, and if there was a difference between the two sites. So I predicted that the cold highland population would age more quickly because it seems like they're investing more in both growth and reproduction. They're growing larger, they're having more babies. Theoretically, that should mean they age more quickly. So to look at the actuarial senescence, I used Bayesian survival trajectory analysis. Don't need to go into all the details, but there was a lot of individuals in the data set, and it allowed me to fit population as a categorical covariate. So I could look at the differences between the populations in mortality and survival, age-specific mortality and survival. And then I fit 4 different models, 3 different shape parameters, and picked the best one based on the AIC. And the best fit for the species was a logistic model with a bathtub shape parameter that looks like this. So we can see that there is positive senescence in both populations here. The mortality increases with age. I've started it at maturity so that we don't take into account the juvenile, which is a bit different. So from maturity, mortality increases, but then it does plateau later in life at around age 5 in both populations, a little bit earlier in the warm population. And the survival starts off high and then decreases down to about 0, but that's survival will be decreasing. And the life expectancy between the two sites is fairly similar, more similar than I was expecting. It's slightly higher in the warm lowland compared to the cool highland. And this is related to the rate of late life mortality in the warm lowland population. It's slightly lower than the cool highland, as you can see here. So there is actuarial senescence, they're aging, but the plateau is interesting. And then to concurrently look at reproductive senescence, I wanted to look at 4 different reproductive traits. So we have litter size and litter mass, which are both measures of absolute investment. And then I used relative litter mass and average offspring mass as a relative measure of investment. So depending on how the female condition, size, everything is, that's what she's investing. Because they have indeterminate growth or asymptotic growth, it's really difficult to separate out age and size in ectotherms generally. So what I did, you can see how closely related size, body size on the x-axis and litter size and litter mass on the y-axis. So I used the growth curve and these to generate residuals to get a size independent age measure of these terms. And I looked for any different shape parameters around linear models, but tested for quadratic effects and interactions, but just straight linear was the best fit for all cases. So what I found was litter mass didn't change with age in either population. They both had sort of negligible effects, and the same for litter size. There's much fewer data points at the end here, of course, because there's not that many lizards that make it to 14, but there appears to be no change with age in the reproductive output. And the same for the relative reproductive output. There's a very small increase with age in both relative litter mass and mean offspring mass. But again, these have very small effect sizes. So they're significant, but it's not really biologically meaningful. And then I looked in detail at the site differences in reproductive output over life. So throughout their life, and on average, the cold highland population individuals have larger litter sizes, they have a higher litter mass, they have higher relative litter mass, higher average offspring mass, and overall higher lifetime reproductive output. So they have more babies over their whole life, and the babies are bigger, and they're bigger compared to the size of the females as well. This is interesting because there's not really a difference in the lifespan. So the cold skinks have something going on that's working for them. They invest more in reproduction and invest more in growth, but it doesn't seem to come at a cost to the cellular maintenance. Because we know from the previous studies that the cold highland population have longer telomeres, and their telomeres slightly increase with age. So they're doing something that's working. And the next steps for me is figuring out what the difference is and how this actually plays out. Could be related to extrinsic mortality rates. The juvenile survival rate is higher in the cool highland population, but only by about 10%. This could be something to do with the actual population characteristics. One of them is very bushy, one of them is open and rocky, and the bushy one is sort of closing in. That's at the warm population. So the population is possibly declining. That might be related to how they're aging and how their life history works. Could also be something related to resource availability. So it would be really interesting to do some little trapping of insects and things at the time where they're active and reproducing. So what does it mean when you take it all together in one big look at aging and life history and senescence and telomeres in the lizards? We do see senescence in the skinks. They're aging. We don't see cellular senescence because the telomeres are actually getting longer as the lizards get older. So we need to have a think about how that fits into evolutionary theories. We haven't seen reproductive senescence. There's no evidence of reproductive senescence as yet. There may be things that I haven't looked at yet that are suffering, but everything looks like it's going fairly well for them with age. There is actuarial senescence, so their mortality does increase as they get older, but then it does plateau at around age 5. So at some point, they just start existing and continuing to exist until they get picked off by a predator. And the life history trade-offs that we expected to see were not present. So either there's something going on that I haven't picked up on, or the ectotherms are playing differently into the life history theory of aging than endotherms. So it's important to look at these differences to think about aging processes across species, across taxa. And as I said, the life history theory of aging is, in short, as individuals grow older, less lifetime fertility remains. So continued survival contributes less and less to reproductive fitness. It is therefore under weaker selection, leading to shorter lifespans. So for the lizards, as they grow older, lifetime fertility does not decrease, it actually increases. They're getting better. So continued survival contributes more and more to reproductive fitness and is therefore under stronger selection, which is maybe why we see some negative senescence in other species. But in the lizards, we don't see negative senescence. So we need to understand how life history theory is predicting this and what the theories of aging can tell us and what the lizards can inform the theories of aging. Another thing that's interesting is the activity of telomerase in ectotherms. They're not seeming to suffer any costs at a cellular level or at an organism level. The current theory for endotherms is that the increased cell division that goes along with telomere repair and continued cell division is there's risks, higher risks of mutations such as cancer. And that's why aging is selected for in endotherms, so that we don't risk those mutations. We just get all the reproduction done early and then decline. But there don't seem to be higher rates of cancer in wild ectotherms. So maybe that theory doesn't hold either. And some more investigation into ectothermic telomere repair is called for, I think. The next steps in my research, I'm still working with the same system. I'm planning to look at individual trade-offs in investment. So looking at the trade-offs between growth and reproduction and maintenance, but at an individual level. So I didn't see anything at the population level, but maybe there's individual decisions being made. So the lizards that invest very heavily in reproduction early on are the ones that die quicker. But we do have some preliminary data that seems to show there's no cost of early maturation on growth or on lifespan. So that will be interesting to see what happens as well. As I said, it would be great to look at some other extrinsic factors. So maybe predation levels and resource availability at the two sites, just to get a broader picture of what's actually going on at the two places. Planning to do some large scale thermal experiments, looking at the effect of temperature on early life changes in telomere length, because early life telomeres predict later life telomeres, which are really important. So seeing what happens early in life, like if there's a big heat wave when they're young, does that affect them long term, or can they cope with that and repair their telomeres? It would also be great, and I plan to get a better understanding of the cellular processes. So doing some more in-depth studies looking at oxidative stress, antioxidant activity, telomerase activity, telomere changes, DNA degradation, mitochondrial activity, all of these things at once in as feasible a way as possible and on different timescales. I'm also planning to catch all of the males at the populations for the next few years as well, so that we can actually generate a pedigree and look at heritability in a more detailed way, and also paternal age effects so that we get a better idea of what's influencing the offspring. So I'd like to thank everyone who's helped make this research possible. Supervisors Eric and Jeff, my PhD, people that were part of the studies, Laura and Adam, Angela and Mats, and funding bodies, Holsworth and Endeavour Awards, and then also to the really supportive and wonderful peer group at UTAS, which of course includes Peter. And in this wonderful and supportive group at UTAS, we have some PhD projects available now. So if you know anyone who would be interested in evolutionary studies, there's 2 potential projects available on this topic. So aging in ectotherms. Could be physiology based, could be molecular, could be more life history. So anyone who's interested, come and talk to me. All right, thank you everyone.
Do we have any questions from the audience?
Thanks. That was really interesting. A lot of things. I just wanted to ask, do you know the body temperatures of your lizards at the 2 sites? And the sort of thing I'm thinking is, particularly if the lowland site is very bushy and there's less basking opportunities, it may be during the active season that they've got higher temperatures at the higher body temperatures of the upland site where there's more rocks and more basking opportunities than the lowland population, because as I'm sure you're aware that in ectotherms, the air temperature can be very different from the body temperature.
Yeah, so that's true. We do know, we've done a lot of basking experiments on the lizards as well. So there's differences in the basking behavior. The cool highline ones do definitely bask more when they're given the opportunity. So they both get to the preferred body temperature, but the cool highline ones just take the opportunity more when it comes up. But when we put them in the basking treatments, we check their temperature with a thermal gun, and it does seem to be that, when I say hot and cold treatments, I'm talking about the availability of light throughout the day. So the cold treatment is 4 hours of light per day, and the warm treatment is 10 hours of light per day. So even if they're getting to their preferred temperature, it's only available for 4 hours, if that makes sense. In the experiment, but in terms of what they actually experience, there have been some studies looking at their metabolic rates at the different sites and when they're given free access, but I think they're around the same.
I actually had still talking about the temperatures and everything. So in the treatments that you did do, was the warm treatment equivalent to the warm temperatures they experience in the warm population, and the cold treatment was equivalent to the temperatures they experience in the cold population, or were they more extreme than those?
The warm temperatures are slightly more extreme and the cold temperatures are slightly more extreme, but they're what the lizards may experience on a bad day at the cold site and a good day at the warm site in summer.
Cool.
Hey, thank you for the talk. It's really interesting topic. I have a question about the tail experiment that you've done. If you can go back to it, did you say that you used the blood?
Yeah.
So do you expect if you use the tissues actually from the original tail and the tissues from the growing tail later, would you expect to have a different results to the blood itself?
Yeah, so I used blood for the main study because it's such a fast cellular turnover, but I did actually plan to do both tissue and blood in this experiment, but there was some issues with keeping the lizards outdoors and the sample sizes dropped so much that I didn't have enough sample size to compare between the regrowing tails and the control tails.
Yeah.
But definitely would have been interesting to see what's actually going on in the tissue.
Yeah. I think you might find more difference with the tissues itself. I do have another kind of clarification. It's not a question. When it comes to the whole process of the telomere in lizards, does it sounds a bit like the telomere in cancer cells? I know you mentioned cancer at the end, but would the processes involved in the telomere lengthening in cancer be similar to the normal telomere in the lizard skin?
Yeah, so there are relations or relationships between telomerase activity, and it's similar to the way that cancer cells grow in that they just keep adding and adding and adding and going out of control. But I'm not across the direct relationships. I haven't done much study into cancer and cell line kind of research, but that's the theory of why we don't do it, is that if we have telomerase activity, it just makes too much risk for mutations like cancers. But the lizards don't seem to have that same problem.
Cool.
Thank you. I'm just wondering, what kind of activity do your lizards exhibit throughout the year? Like, are they active throughout the entire winter, or is there any difference between the two populations?
Yeah, so there are slight differences between the populations, and that's something else I'd like to look at, is the effect of winter temperatures and hibernation on the two populations. So at the cold highland, they go down slightly earlier for their brumation. They're under a blanket of snow. They're not getting woken up. If there's a warm day, they're just staying down there. Whereas the warm population in the lowland, they're not as deep down, so they're not as sheltered from temperature fluctuations in winter. And there are more days that are at their active body temperature. So it's possible that their metabolic rate is rising throughout winter, which is costly when they're brumating because they're not able to go out and get resources. They're just using more resources. So there's been some studies by my colleagues in Sweden about the effect of winter temperatures on the lizards after brumation.
Mm-hmm.
And they found that, yeah, warmer temperatures are costly.
Thank you.
Any other questions from the audience?
Thanks for the great talk. Just clarification. This lizards, obviously, you have two populations, and one obvious question would be, why not have in 17 in between? So is the distribution of the lizard, is it 100% kind of guaranteed that it's one species and not kind of a subspecies because they're pretty far apart? So do they occur all in between, or is it?
Yeah, not across the whole population, but there are definitely populations at different elevations around Tasmania, and they are definitely the same species. There's some closely related species that are alpine specialists and some that are more generalist lowlands, but this species, the ocellatus, do occur, all around, which is quite interesting. And they have these life history differences, but they're not actually far enough apart to be different. Peter's done a lot of genetic work on them, so she might be the person to ask about their how closely related the two sites actually are.
Thanks.
Questions in the chat. So unless there's any other questions, please join me in thanking Louisa for a really interesting talk.
I think I went way too quick then. Sorry, that's a lot. Yeah.